Now we're going to talk about Chapter 3, Biology and Behavior. How does the nervous system operate? Well, it consists of a central nervous system and a peripheral nervous system. The central nervous system consists of the brain and the spinal cord, whereas the peripheral nervous system consists of all nerve cells in the body that are not a part of the central nervous system. The peripheral nervous system includes the somatic and autonomic nervous systems. Here's a diagram to make that make sense. Neurons are the basic units of the nervous system. The nervous system is responsible for everything we think, feel, and do. Its basic components are neurons. These are basic units of the nervous system and are cells that receive, integrate, and transmit information in the nervous system. They operate through electrical impulses, communicate with other neurons through chemical signals, and form neural networks. Nerve cells are powered by electrical impulses and communicate with other nerve cells through chemical signals. There are three basic phases: reception, integration, and transmission. Reception: chemical signals are received from neighboring neurons. Integration is when incoming signals are assessed, and transmission occurs when signals are passed on to other receiving neurons. Here is a close-up picture of a neuron, and I'll say more about that here in a second. There are also sensory neurons, motor neurons, and interneurons. Sensory neurons are neurons that detect information from the physical world and pass that information on to the brain. Somatosensory nerves provide information from the skin and muscles. Motor neurons, these are neurons that direct muscles to contract or relax, thereby producing movement. Interneurons are neurons that communicate within local or short-distance circuits. Now we're going to discuss the structure of a neuron. So tie back into that picture that I showed you. It consists of a dendrite, a cell body, and an axon. The dendrite are the branch-like extensions of the neuron that detect information from other neurons. The cell body, or soma, is the site in the neuron where information from thousands of other neurons is collected and integrated. The axon is a long, narrow outgrowth of a neuron by which information is conducted from the cell of the body to the terminal buttons. And to go back to that image, up at the top, the branch-like features up there are the dendrites. The bright white area in the middle of that is the cell body, and the area protruding out of the bottom of the cell body is the axon. Here's a cleaner picture of basically the same thing. Messages are received by the dendrites on the far left, and they're processed in the cell body, and then transmitted along the axon and sent to other neurons via chemical substances released from the terminal buttons across the synapse. The synapse is that space between the terminal buttons and the next neuron. To reiterate, terminal buttons, those are at the end of the axons. They are small nodules that release chemical signals from the neuron into the synapse. The synapse itself is that gap, as I mentioned, between the axon of a sending neuron and the dendrites of a receiving neuron. This is the site at which chemical communication occurs between neurons. Show you the diagram there again, just to see how the information flows along. Alright, now we're going to talk about action potentials. These are what produce neural communication. An action potential, also known as neural firing, this is the electrical signal that passes along the axon and subsequently causes the release of chemicals from the terminal buttons into the synapse. A resting membrane potential. This is the electrical charge of a neuron when it is not active, i.e. resting. It can become polarized, and this is when a neuron has more negative ions inside it than outside. I'll say more on that here in a second. Polarization creates the electrical energy necessary to power the firing of the neuron. Without that electrical charge, the neuron will not fire. Alright, now I'll discuss those ions that help facilitate this whole thing. The sodium ions and potassium ions contribute to a neuron's resting membrane potential. Ions pass through cell membrane channels, and this flow of ions through each channel is controlled by a gating mechanism. That gating mechanism is called the sodium-potassium pump. This is a mechanism in the membrane that contributes to polarization. It increases potassium outside the neuron and decreases sodium inside the neuron. All right, here's a diagram of resting membrane potential. A neuron is at rest. It is polarized. It has more negative electrical charge inside than outside. The passage of negative and positive ions inside and outside the membrane is regulated by ion channels, such as those located at the nodes of Ranvier. Study this diagram here to see what's going on. Alright, now it's changes in this electrical potential that lead to action. Neurons receive chemical signals from nearby neurons. These signals can either be excitatory or inhibitory. Excitatory signals depolarize the cell membrane, i.e. reduce polarization, increasing the likelihood that the neuron will fire. It gets excited. Inhibitory signals, however, hyperpolarize the cell, i.e. increase polarization, decreasing the likelihood that the neuron will fire. It inhibits the firing. Okay, and that leads to either a firing state or a resting state. These are not terms in the book, but I think it organizes the information better. When a neuron fires, the inside of the neuron becomes slightly more positively charged than the outside. After firing, the inside of the neuron returns to its slightly negative resting state. Now here's another diagram that focuses just on the electrical component of this. This is an action potential. The electrical charge inside the neuron starts out slightly negative, which is the resting membrane potential. To be exact, it is negative 70 millivolts. As the neuron fires, it allows more positive ions inside the cell, process of depolarization. It then returns to its slightly negative resting state. This happens at each portion of the exposed axon as the action potential travels down the axon. Action potential spread along the axon. Depolarization travels along an axon like a wave. Action potentials always move away from the cell body to the terminal buttons. There's something called a myelin sheath. It is a fatty material made up of glial cells that insulates some axons to allow for faster movement of electrical impulses along the axon. The nodes of Ranvier, as I mentioned earlier, are small gaps of exposed axon between the segments of myelin sheath, where action potentials take place. The all or none principle. Neuronal firing is determined by the number and frequency of signals the neuron receives. An action potential occurs when the sum of excitatory or inhibitory signals leads to a positive change in voltage that exceeds the neuron's firing threshold. This leads us to the all or none principle. This is the principle that when a neuron fires, it fires with the same potency each time. A neuron either fires or it does not. It cannot partially fire, although the frequency of firing can vary. It either receives a signal that's strong enough to trigger the firing, or it does not receive a signal strong enough to trigger firing. Neurotransmitters influence mental activity and behavior. What is a neurotransmitter? You may have heard of this term before. This is the chemical substance that transmits signals from one neuron to another. This occurs in the synapse from the terminal buttons. There are also receptors. In neurons, specialized protein molecules on the postsynaptic membrane, i.e. the receiving neuron. Here, neurotransmitters bind to these molecules after passing across the synapse. The binding of a neurotransmitter with a receptor produces an excitatory or inhibitory signal. Here's a close-up diagram of a terminal button and a synapse. Should study this diagram. This shows you how neurotransmitters work. The first step is neurotransmitters are made inside the axon. Then neurotransmitters are stored in vesicles. Action potentials causes the vesicle to fuse to the presynaptic membrane and release their contents into the synapse. Here, the released neurotransmitters bind to the postsynaptic receptors. Neurotransmission is terminated by reuptake, enzyme deactivation, or autoreception. If you've heard the term reuptake before, it's part of an SSRI. It is an antidepressant. A selective serotonin reuptake inhibitor causes for there to be more serotonin left over in the synapse, thus increasing the amount of serotonin, which they believe leads to greater happiness. Neurotransmitters bind with specific receptors. Neurotransmitters stimulate specific receptors and block new signals until terminated. The three major events that terminate the neurotransmitter's influence in the synapse, as I was just discussing, are reuptake, enzyme deactivation, and autoreception. Reuptake means the neurotransmitter is reabsorbed into the presynaptic terminal buttons from where it came. Enzyme deactivation, an enzyme is secreted and it destroys the neurotransmitter. It's kind of like recycling. Autoreception signals the presynaptic neuron to stop releasing the neurotransmitter. All right, now we have to talk about agonists and antagonists. Drugs and toxins can alter neurotransmitter action. Agonists enhance the actions of neurotransmitters. Antagonists inhibit the actions of neurotransmitters. So some drugs and toxins can act as agonists or antagonists in terms of neurotransmitters. Researchers often inject agonists or antagonists into animals' brains to assess how neurotransmitters affect behavior. All right, here's a diagram that explains agonists and antagonists. Agonist drugs can increase how much a neurotransmitter is made so that there's more inside the vesicle. They can also block the reuptake of a neurotransmitter, which will then also increase the neurotransmitter. An antagonist is a drug that can decrease the amount of the neurotransmitter so that there is less inside each vesicle. They can also help destroy the neurotransmitters in the synapse. Now we're going to move on and discuss the different types of neurotransmitters. The first one is acetylcholine, ACH. This is a neurotransmitter responsible for motor control at the junction between nerves and muscles. It is also involved in mental processes such as learning, memory, sleeping, and dreaming. Nicotine, chemical that's in cigarettes and certain vape products, imitates this neurotransmitter and binds to those receptors. Acetylcholine and Botox. Acetylcholine is responsible for motor control between nerves and muscles. Botox inhibits the release of acetylcholine, thus paralyzing muscles. Here a woman receives a Botox injection to remove wrinkles in her forehead. It works because of acetylcholine. Other types of neurotransmitters, ones you may have heard before, are norepinephrine, serotonin, and dopamine. They are grouped together because each has the same basic molecular structure. Norepinephrine is a monoamine neurotransmitter involved in states of arousal and attention. Serotonin is a monoamine neurotransmitter important for a wide range of psychological activity, including emotional states, impulse control, and dreaming. And dopamine is a monoamine neurotransmitter involved in motivation, reward, and motor control over voluntary movement. Lesser known types of neurotransmitters include GABA, glutamate, and endorphins. GABA, also known as gamma-aminobutyric acid, is the primary inhibitory transmitter in the nervous system. Glutamate is the primary excitatory transmitter in the nervous system. And endorphins are neurotransmitters involved in natural pain reduction and reward. It is said that joggers, when they get back from a long jog, experience the relief of endorphins after that, and actually creates pleasure after exercising. Here's a diagram from the book that groups them all together, gives you psychological functions for each. It will help organize this information for you. You can pause it here and study this, or crack open the book or ebook and get at this image. Just helps organize the information. And thus concludes part 1 of chapter 3. Be sure to listen to the second part of